Hi, good morning, everyone. Thank you for joining us. I'm Heidi Zimmerman, Executive Director at AWT. And just a quick housekeeping note, when you signed in, you'll notice you were in listen-only mode, but we do encourage questions. So as you have them, you can go ahead and just type those in the chat box, and we'll take those certainly at the end, if not before. And also, this webinar is being recorded, and a copy of the webinar will be posted on the AWT website, and a PDF copy of the slides will be sent out to all the attendees as well. So with that, let me introduce our speaker today. Dr. Dan Broder is team leader for new product development within the R&D division of IDEXX's water line of business, where he and the R&D team develop new methods for quantifying waterborne microorganisms. He is the project lead for Legiolert, IDEXX's new quantitative method for detecting Legionella in potable and non-potable water samples. Dan received his PhD in microbiology at the University of Illinois at Urbana-Champaign. So at this point, let me turn the program over to Dan.
Thank you, Heidi, and welcome everybody. So today's webinar is Reducing Legionella Risk with Confidence. And let me just give you a brief agenda as to what I'm going to talk about. And again, I would invite you to, if you have any questions, you go ahead and submit them, and we'll try and address them at the end of the webinar. So today I'm going to talk about, I'll give you an introduction into Legionella, though I think probably by the fact that you've tuned in, you probably have a pretty good idea of what Legionella is. But for completeness, I'll talk a little bit about it, where it lives, and how it causes illness, and why there is a concern for Legionella. And then I'm going to go and talk a little bit about how you can control it and how you can manage the risks associated with Legionella and Legionnaires disease. And then I'll talk a little bit about the responsibilities, where the responsibilities lie and where the liabilities exist. And then I'll talk a little bit more about something that's a little bit closer to my heart, which is the methods that we use for detecting Legionella in water systems and how those work in conjunction with any good water management plan. So, next slide. Oops. Okay. So what is Legionella? As you probably know, it's a bacterium that was associated with an outbreak, a mysterious outbreak that happened in 1976 at the Bellevue Stratford Hotel in Philadelphia. And what ended up happening was there was about 2,000 attendees at the American Legion convention, and a little over 200 people got ill and 34 people died. And they didn't know exactly why that happened, but ultimately tracked it back to this organism that they later named after the event and called it Legionella. Now, Legionella is not actually one organism, it's a group of different organisms, but they've sort of collectively termed them Legionella. In actuality, it is, I'm just trying to change my slides here. It's actually a group of different organisms. The primary one that you hear about is Legionella pneumophila. This is far and away the most important species of Legionella. It's the one that's most commonly found in the environment, and it's the one that's nearly always associated with disease. Legionella pneumophila can be further subdivided into what are called serogroups or serotypes, I will sometimes call them, which is really just a way to describe different surface structures that live on the outside of the different species of Legionella. And there's upwards of 15 different serogroups. The one that seems to be most associated with outbreaks, and the one that was characterized by the original outbreak, is serotype number 1. There are upwards of 15 of those, but 1 is the one that seems to be the most pathogenic by and large. The other ones do have pathogenicity associated with them as well. There are other species of Legionella out there. Last I heard, there was about 50 of them. Most of them are not associated with disease, but there are a handful that have been associated with disease on rare occasions. But these ones exist out in water systems, albeit to a much lower frequency and much lower incidence. Regardless, Legionella has a lifestyle that is pretty ubiquitous in water systems. But the real problem exists with things like biofilms. What is a biofilm? A biofilm is a stable structure that can act as a breeding ground and dissemination mechanism for microorganisms. And the basic way it works is pretty simple. A microorganism can float around in a fluid matrix, and it can attach to a surface. And when it does, it can secrete different compounds and molecules that will form kind of a layer around it, protect it and keep it adhered to whatever surface it binds to. Now, these things can be communities. They can exist. They're usually not just one organism. They're usually a whole community of a lot of different organisms. And some organisms that don't naturally form a biofilm can associate with a pre-existing biofilm as well. But if you have one, you're going to get a pretty good enrichment of whatever organisms are living in there. And this process follows where they grow up to a high number and they can be released back into the environment, where they either can form biofilms in new locations, or they can just float out in the water and come out. In the worst case scenario, they'll come out of your tap. And if you look at a cross section of this pipe below, you can see what it might look like. On the left is sort of a hypothetical drawing of how a biofilm can occlude the inside of the pipe, and if that's a high concentration of organisms that can float out into the water. And the one on the right, on the bottom there, is a real cross section of a pipe, and that's probably a worst case scenario. You can see that it's nearly almost closed up. But you can imagine if that's a pipe that's leading into your building, you wouldn't really want that to be the case. And you can imagine there'd be a high number of microorganisms that might slough off into your water and come out into your system. Legionella live in a lot of different water systems, as I said. One, I think, underappreciated place they live is in potable water. I know when people hear Legionnaires disease, they think of cooling towers, and that is definitely true. But the fact is that potable water is just as dangerous, if not more dangerous, of a location for Legionella to reside and to multiply within as it relates to Legionnaire's disease. And potable water can be anything that's exactly that. Potable means, can you drink it? It can be showers, taps, sinks, faucets, ice machines, humidifiers, anything that you might drink or ingest in any way can be classified as a potable water. They also live, of course, in non-potable waters, and that is a pretty broad group. In general, non-potable waters are cooling towers, which is depicted in the central two images here. The top is just sort of a zoom out, and on the bottom part is the fan that actually blows the aerosolized Legionella water droplets into the environment, or the humidity into the environment. But there can be fountains, decorative fountains can be sources, spas, baths, chillers, even misters in the supermarket can all be roughly classified as non-potable water, and Legionella can live in any of these. So how does this work in terms of Legionnaire's disease? Well, if you look at the image on the left here, you can see in a shower, if you have a biofilm that's living somewhere in the pipe, and this image is depicted right before the shower head, if there is a biofilm living there and Legionella can slough off that into its planktonic or swimming form, then it can blow out the shower head and it comes out in an aerosol. It's that aerosol that's really the problem if it contains Legionella or Pneumophila in it. Likewise, if you look at a cooling tower, this is sort of a complicated diagram of a cooling tower, but in essence, what a cooling tower is a big fluid reservoir and a fill material that is served to cool off the water so it can be shuttled back into a building and cool off the heated building elements. But at the top is a big fan, and basically what the fan does is it cools off the system by blowing the humid air out of it. And in that humid air, if there's Legionella in the reservoir or in the fill material, that can blow out into the environment. Now, cooling towers can be massive, and those fans can be huge. And if that's the case, and you have a lot of Legionella pneumophila in that system, you can imagine that it might blow a huge amount of that aerosol out into the environment, which is probably why a lot of the big outbreaks tend to be associated with cooling towers, because they blow out so much of this stuff out into the environment, and it can go a pretty significant distance. So what is the importance of aerosols? The aerosols are important because that's how you get Legionnaire's disease. Legionnaire's disease is not something you get from blood to blood or person to person. You don't really get it from another person. You get it from breathing in infected water droplets into your lungs. And for that reason, there are certain risk groups that are higher at risk for Legionnaire's disease. People who have chronic lung disease or are smokers, immunocompromised people, or elderly people are all at higher risk, and it's precisely for that reason. It's because of the mechanism of how Legionnaire's disease is contracted. Now, that is one risk factor, is the person, the susceptibility of the patient. There's also the virulence of the strain, which I mentioned before. Legionella pneumophila serotype 1 tends to be the most virulent, so if you are a susceptible person and you have been exposed to a very virulent strain, then you have a lot of risk factors. There's also the extent of the exposure. How small were the water droplets? It tends to be the case that the smaller they are, the worse it is, because you have a higher tendency to breathe them in. Or how close were you to the source of where those aerosols were produced, which is going to be a reflection of the dose that you get when you breathe it. Now, the interesting thing about Legionella pneumophila is that it's not actually a terribly good pathogen. It's not really very good at what it does. It kind of has to have all these things align in order for you to get ill. If you have 2 of the 3 risk factors, you may still get sick, but odds are lower. You kind of have to have all 3. And for that reason, it's something that you can deal with. And this is sort of the overlying theme of this webinar is how do you deal with this problem? And it turns out there's a lot of great ways to deal with it. You just have to be diligent. So, should you even be concerned with this, given that's the case? The answer is yes. The number of cases of Legionellosis has gone up over the past 14 years. I pulled this plot on the left here right off of the CDC website, and there's the reference there. You can look at the data for yourself as well, but it just keeps going up and up over time. Some of this is due to increased reporting, but not all of it. Some of it is truly an increase in incidence. Between 8,000 and 18,000 people get Legionellosis each year in the US, and about 10% of those are fatal. Now, one other interesting thing about Legionnaire's disease is if you look at some other sources of infection of with Legionellosis, you can see one particular source I like going to is the European Center for Disease Protection and Control. They put out an annual report, a surveillance report, which has all kinds of details and parameters and data about Legionellosis. And one thing you see in each report is the seasonal incidence of Legionnaire's disease. And you can see pretty clear, this plot changes subtly from year to year, but it's basically the same every time in that there's always this spike from the July to October area, which is the warmest season. And I can't help but think that the seasonality of Legiolert associated with climate change and the fact that everything is getting just a little bit warmer is going to change the shape of this over time, in that the peak is going to get a little bit higher and the shoulder is going to get a little bit broader. And the overall output of that is you're going to get more overall Legionellosis each year because of that trend. Some significant Legionella events in the last 16 years. And I don't want to go through each of these. I just want to show you some notable things and some notable trends. In particular, you have some big ones that I've bolded here. The cooling tower outbreak in Quebec City. I'm going to use my mouse here to show you where that is. There was 180 people that came down with Legionnaire's disease and 13 people died. There, of course, is the ongoing problem in Flint, still not over, which was the lead originally, but actually there's a Legionnaire's disease problem there as well. 91 people so far and 12 people have died. And then, of course, the outbreak in the South Bronx associated with the cooling tower where 130 cases were shown and 12 people died. Now, with the South Bronx event, that actually stimulated some legislation. Now, every cooling tower has to be registered and tested, I believe, on a quarterly basis. But what's interesting about this data table is that if you look, there's a lot of these that are potable. And though they're not outbreaks in the traditional sense, in that lots and lots of people are not all simultaneously getting sick at the same time, there are a lot of deaths associated with this. And this is sort of the thing I was alluding to before, that potable water has not gotten as much notoriety for being a problem, but the fact is that it is a problem. And if you look at the same CDC data set or website or report from Morbidity Mortality Weekly Report, you can see that a significant number, if not most of the outbreaks and confirmed and suspected cases are the result of potable water, if not more than cooling towers. So Legionella, as I said, is a ubiquitous organism. It's in most water systems. Up to 70% of building water systems have Legionella at some level in them. Your job is not to get rid of it. You can't really eradicate Legionella. What you can do is control it, and that's really the theme here, is control the water system with a good risk management plan. And the CDC has estimated that out of every 10 cases, about 9 of them could have been prevented with good water management. There are tools to having a good water management plan. In 2015, the American Society for Heating, Refrigeration, and Air Conditioning Engineers, also called ASHRAE, came out with their standard 188P, which helps you understand what you can do to reduce the risk of exposure to Legionella bacteria. And it tells you, it guides you on how to do a risk assessment of a building. It's got a lot of details in it. It's got a recommended water safety plan. got a section that talks about how to do the testing, and it's really the first North American standard for Legionella. It's a really useful tool. It's not the only tool out there. There's also the CDC Legionella toolkit. This is a very, very easy to follow step-by-step instructional guide to how to set up a risk management program. It's got a worksheet, just a yes/no kind of worksheet to examine a risky building area. It walks you through a standard Legionella management program. It's got some examples of what might be a problem scenario and what a person might do to deal with those scenarios. And it even has guidance for specific facilities. You can imagine if you have a nursing home or a hospital where you have a much higher population that's at risk, those might require specific attention that's different than perhaps a gym or something like that. So this walks you through all those kinds of things. It tells you how to do critical control points of the building system so you know where the most important testing locations are within the building. And it has other details about how to manage your biocide treatment and things like that. It's pretty thorough. So there's tools out there, but by and large, the one thing you want to realize is that when you do any kind of risk management plan or water management plan, it's one thing to have your plan and to have a team that's designed to execute it. But one key part about that is to have a plan to test to make sure that it's working. And a key part of that is testing to determine whether or not the amount of Legionella in your system has actually gone down or is being maintained at a low level so that safety is ensured. Now, when you do testing, you need to make sure that you have reliable results so that you know that whatever concentration of Legionella you have in your system is acceptable. What is acceptable? Well, it depends who you ask. A lot of the entities out there have guidance limits for what concentration of Legionella pneumophila you can have in your system. And they're all roughly similar to each other. There is some differences, but they're all pretty close to each other in terms of what is acceptable. For the CDC and for ASHRAE for potable water, it depends on whatever your risk management plan is, which depends on the building and the whole setup of the building. Entities like AIHA or OSHA all suggest that you should have about 10 CFU per milliliter of bacteria. That's the CFU stands for colony forming unit and is usually equated to actual cells of bacteria. And then for the Veterans Administration, they basically say any positive. And for places like the New York State Department of Health, they take a slightly different approach, which is to determine whether or not a percentage of the total outlets in your building have a positive output. So they all kind of differ, but they all are basically the same in that they're determining whether some minimum level is being exceeded. For some countries like France and Germany, they actually have legislation. You have to test and you have to meet a minimum requirement in order for your water to be considered acceptable. That's for potable water. For non-potable water, it's similar. You basically multiply everything by 10. Potable water tends to have a higher overall bioload or background organism content, so the limits are a little bit more forgiving by and large. What does this mean functionally speaking? If you breach one of these limits, then there's some sort of action that you need to take. And those actions differ based on who you ask, but again, they're all fairly similar to each other. And this is an example from AIHA, which is the American Industrial Hygiene Association. You can see that there's several different levels, and they're pretty verbose into what they suggest you might do. But the basic idea is if you're at some minimum level, you're below the action, then you don't really do anything. You just continue monitoring to make sure that what you're doing is continuing to work. If you have a concentration that exceeds one of these designed or guidance limits or legislative limits, then you need to take action. Usually the first action is to retest to make sure that your first test was correct. And if it was, then you do something about it, which is typically an increase in the biocides, chlorine or chloramine or copper silver or whatever your disinfection method of choice is. And then you test it again to make sure that that biocide increase has had the desired effect. If you are perhaps 100-fold higher than the action limit, then the actions become a little bit more dramatic. You might shock the system with a high amount of chlorine, or you might boil the water. And if it's even higher than that, then you get into this outbreak scenario where you probably would shut down the building or sequester people from going into certain parts of the building that are higher risk, things like that. So a lot of these kinds of things are based on regulations or guidance. Regulations are less common. They are on the rise, and they tend to increase whenever there's bad press. Like a good example of that would be the Bronx outbreak. There were no regulations prior to that, and when it happened, they stimulated some regulations that required the registering and testing of cooling towers and healthcare-associated facilities. Guidance is more common. It's well known that conforming to guidance is going to reduce the risk of illness. And not conforming to guidance can, of course, result in illness, which may result in fatalities, which is bad, but it can also result in liability. Who is to blame? Generally, the owner or operator of a public building will be to blame. Building engineers and maintenance personnel may also be to blame, depending on the specific circumstances. I was reading recently about a lawsuit where there was a cruise line where some people got ill and died with Legionnaire's disease, but the cruise line itself actually sued the manufacturer of the sump pump, and they won the lawsuit because the sump pump manufacturer made them in such a way that it wasn't designed to properly control for things like Legionella in the system. So it's pretty interesting, and it can get pretty creative who's to blame, and it's significant. There is a big impact of the liability. And I did a little exercise before this webinar where I just went to Google and I typed in Legionella lawyer, and there's a lot of hits. And I just took those hits and I mapped them out by location to the United States. And after I got to about the 6th page of hits, I kind of quit. So this is not a comprehensive list. But I mapped them out, and you can see there's quite a few all over the United States with some geographic gaps, probably just because I didn't go all the way to the 20th Google hit page. But in general, there's a lot of lawyers out there that either specialize completely in Legionella cases or have Legionella-related cases as part of their practice. People do sue for getting Legionnaire's disease. The settlements and the awards can be significant. This is just a snapshot of some of the more significant awards and settlements that have happened in the last 10-year period. There's a lot more than this. But what is not encapsulated in this table is the legal fees. Maybe if someone gets sick and you did everything you're supposed to, and you did basically what you're supposed to do, then you still may have to go to court. And if you go to court, it's going to cost you to pay for a lawyer. Even if you win, it's still going to cost money. The best and most cost-effective thing to do is just to make sure that nobody gets sick by having a really, really solid risk management plan. That's the best way to do it. So, test regularly to confirm your risk management practices are working properly. If you do that, then you will be enabled to perform corrective actions whenever they're needed, and then retest to confirm that those actions have actually reduced the risk. And then keep records of everything you do, because if God forbid something happens and someone does get ill and there's some sort of litigation that results from that, you have all of the proper paperwork and results that show that you did what you're supposed to do. And sometimes biology just happens and disease just happens, but there's things you can do to control it. So now that brings me to the other part of my talk, which has to do with testing. Of course, testing is the way to confirm your risk management practices are working, so it's an important part of your plan. What do most labs do for testing? The truth is that most labs do culture. It's the gold standard. It is the one that tends to be very, very reliable. It's the one that people have the most experience with. It's been tried and tested over time since basically the beginning. And what you're looking at here is a Petri plate. This is a black Petri plate called BCYE. It's commonly used for culturing Legionella species. And you can see here, this is basically what Legionella looks like on the plate. These are colonies or colony-forming units. You can actually see there's more than one type of Legionella on this plate if you look closely, which is part of the point I'm going to make, is that there's definitely an expertise level to looking at Legionella plates. Now, the problem with culture is that it's not one method. The culture method is a bunch of different methods, and people just kind of pick and choose whatever they want to do. There's a lot of different protocol options. Some people will concentrate their sample, which you can do with membrane filtration or with centrifugation. Some people will do pretreatments where they'll treat it with an acid or a heat step. Legionella has some really strange resistances. It's resistant to low levels of acid and heat, where other microorganisms may not be. People will choose a variety of different selective formulations. They'll put antibiotics on top of their BCYE plates that will prevent the growth of the background organisms. And there's a lot of different choices of antibiotic formulas that you can use. This is just a few of them. And people just select whatever they want to do. And then whenever you're you've done your test and you look at these colonies here, you're not done just by looking at them, because you can't definitively say that one is Legionella or is not. You can be very suspicious that they are, but you're not done there. You have to do some sort of follow-up and confirmation method to make sure that it actually is Legionella if you think it's Legionella. And there's a lot of options for doing that as well. So there's a lot of different methods out there, which means there's a lot of different results. And I'm not going to go through this in detail. These are just snapshots of diagrams of some of the existing methods out there. The ISO method that's used in Europe is on the top left, the CDC method that's used domestically, and then the AFNOR method that's used in France and some regions in Canada are all methods that are used to detect and quantify Legionella. You can see they're all sort of similar. They're also pretty different. Some of them use different selective formulas, some of them use different steps, some of them use different numbers, total numbers of plates. But in each case, what they're going to be doing is running multiple different conditions in parallel, all on the same sample, because they don't know which one's going to be the right one to give them the correct result. Whichever ones don't work, they'll just throw in the trash. Whichever ones look like they might work, they'll move forward with their normal confirmatory work. And when they get the plate that they like the best, that's the one that they're going to use for their final result. So they're all pretty different from each other, and there's a subjectivity associated with that because the user has to have the expertise to make the selection as to which result is the one they like the best. And then they might do different confirmatory work at the end as well. And this is just a snapshot on the bottom right of, this is right out of the CDC protocol, just an example of what kinds of results you might expect to be seeing on these plates. And you can see that some of them have that grayish Legionella looking colony and some of them don't. So as the user, you're looking at all these plates and trying to make a decision as to which one you think is the right result, and that's the one that you move forward with. So that's the culture method. It's not a fast method. It takes about 7 days for the initial colonies to come up, and then you got to do confirmation work from that if you get a positive result. So you're somewhere in the 10-day range usually. It's quantitative, but it's very labor intensive and it's subjective, and there's a high variability from method variations and expertise. Now, a lot of other people have come out with what are so-called rapid tests, typically considered to be same day. The biggest one is PCR. PCR is fast. It's kind of quantitative, but it reports out in genomic units, which are not viable cell units. They're not equivalent to cell-forming units or most probable number, which I'll talk about in a minute. It can't tell you the difference between live and dead cells. It's just looking for nucleic acids, DNA, or RNA in some cases. So if you have a building that had Legionella before, and now the Legionella are gone or have been properly controlled, and you do PCR, then you may still get a positive result because it's just looking for the DNA. The DNA doesn't necessarily go away. It can also suffer from cross-reaction from other chemicals in the water, which we call the matrix. There's also direct fluorescent antibody microscopy, which is fast, but it's not really reproducibly quantifiable. It also can't tell you live versus dead. There's a lateral flow antibody test, which is kind of like a pregnancy test. It is very limited in, it's not sensitive, it has a high false negative range, and it can't really test very many different strains. And then a really interesting one is antibody capture, which is fast and can be quantitative, though the accuracy does vary. Problem with this guy is that it's also very, very labor intensive, and it's not really practical for a routine monitoring, risk management plan monitoring kind of scenario. So none of these things really match culture in terms of its utility, but they're all touted as being fast. But the problem is, do you really need a same-day test result? The answer is no, not for routine monitoring. The key to a risk management plan or a water management plan is trending, exposing a problem over time. Most water management plans function by testing possibly every month or quarterly. So if you're waiting to test quarterly, the need to have a result the same day doesn't really seem like it's practical. What you really just need is some sort of test that's going to tell you whether your plan is working. Same-day tests are good for outbreaks. If there is an outbreak that's made the news and a lot of people are sick, then I think time is definitely of the essence. And take, for example, the New York State Department of Health. The way they do it is if there's an outbreak, they'll take their samples and they'll PCR test them. PCR is typically going to be more sensitive in that it'll pick up live or dead cells. So if they have a positive, then they will go ahead and do a culture. Even if the result is positive, do a culture anyway for two reasons. One is they need to get a quantification and understand what is the real concentration of the organism in that sample. They also want to take the bug home and stick it in the freezer, because if someone comes into the hospital and they culture from that person, it ends up being Legionella pneumophila serotype 3. They want to see if serotype 3 is the thing they actually collected from the cooling tower on 5th Avenue. So if they get a negative, then they just stop there and they don't do any culturing. So they still rely on culture to do the real bulk of the scientific data work. The PCR part is just to react quickly if they think that there's a reason that would be beneficial. So for routine testing, the best decisions come from data that's accurate, that's highly reproducible. It needs to be reproducible so that the trending is accurate. If you're doing trending and you have some anomalous, massively high data point, you want to be able to know if that's real or not. If you want to treat the system, you want to be treating it based on the proper information. So the trending is accurate no matter who is processing your sample or where the sample is being processed. And you want something that is specific for the disease-causing agent, which is Legionella pneumophila. So that's a nice segue for me to talk about Legiolert, which is really my area of expertise, the testing methods, and Legiolert is what came out of that. Legiolert is the next generation culture test. It is ideal for routine monitoring and for compliance for the following reasons. It detects all serogroups of Legionella pneumophila. It's very specific. There's very little background interference. It's very, very simple to use, and this is the key part of this test, is it's very simple to use. It's just a color reaction. And if you've ever used any IDEXX products like CoLalert or SuitAlert or EnterAlert, it's very similar to that. It's just a binary on-off decision. It's very reproducible and repeatable because of that. It can be used for any kind of relevant water matrices for which Legionella is normally found. It utilizes most probable number, which is an algorithm that is basically the same as cell-forming units. There is no functional scientific difference between MPN and CFU. We get this a lot. CoLalert's been around for over 20 years, and it's been used, and MPN is the way that CoLalert uses its quantification. It's exactly the same as that. And the Legiolert test can count up to 2,300 MPN per test. A Petri plate is pretty much maxed out at about 300. So this is almost 10-fold higher, and it's more reliable. It incubates for 7 days, so it's not a same-day test, but it is faster than the culture because you don't have to do any confirmation work. Once you get to 7 days, you can read the tray, and it's done, and your result is your final result. Now, this is a this is a culture. So what you're looking at here is what we call the Kewanee Tray. The Kewanee Tray is really just a device that holds fluid in it. It holds the culture. And these end up being live cell cultures, liquid cultures in there. And this is a real result from an actual sample. You can see Legionella pneumophila is growing by the fact that the color turns brown in the tray. So all you got to do is go in and count the ones that are brown and total those up and put those into a lookup table, and it tells you what the concentration is. There's really no subjectivity to it. You don't have to discriminate whether whether you think the colony is Legionella pneumophila or not. You just count whether it's brown or not, and it counts all serotypes of Legionella pneumophila. Now this thing works in conjunction with a powdered blister pack reagent in the following way. We have two different protocols, one for potable and one for non-potable water, and that's it. There's no other decision making that a user needs to make. Protocols are fixed. And they're very similar to each other with a couple of key differences. Non-potable water, in both cases you would add the reagent to a sterile diluent. For non-potable it's 100 mils of diluent, for potable it's 90 mils of diluent. For the non-potable test, the water sample would be treated for a minute. An equivalent amount of water sample is added to the same volume of pre-treatment reagent, which comes with the Legiolert kit. You mix that together, cook it for a minute, and drop it into the water reagent mixture, and then pour it into the Kewanee Tray. And the way this thing works is the Kewanee Tray is really like a bag that's sealed on 3 sides. And after you get your sample and reagent mixture, you pour it in there, and then you send it through this device called a Kewanee Tray sealer. And this heat seals the fluid so that it compartmentalizes into all the different wells here. And this thing has 6 large wells and 90 small wells. So you get a lot of volume capability, but you also get really good resolution from these really tiny small wells. And once it goes through the heat sealer, it's done. All you got to do is stick it in the incubator and cook it for 7 days. And when it comes out, it's a confirmed result. All you got to do is count up the number of positives. The potable test is exactly the same, except you don't have to do the pre-treatment step. You just add 10 mils of water to your 90 mils of diluent with the powder reagent in there. You shake that up until it's dissolved, put it in the Kewanee tray, and also seal it. The other key difference is there's an incubation temperature, and this has to do with the selectivity component of the tests. But other than that, they're exactly the same. The output is the same. The detection range is the same, and the quantification is the same. So, does this work? The answer is yes. We've done a lot of different studies to try and understand the performance of Legiolert. One of the first things we did was a 13843, which is a method validation. It compares the method against itself to understand how well the normal user parameters function. And all of these things have a mathematical basis to them, but basically the sensitivity and specificity are ways to measure whether or not, of all the positives that exist in your sample, how many did you find, and of all the negatives that existed, how many did you properly assign. Things like false positive measure how many signals that were positive came up were actually Legionella pneumophila, and vice versa for false negative rate. Now, the repeatability and reproducibility is really one of the key features about this study. Repeatability measures if you have a user, and let's say they have 100 Kewanee trays in front of them, and they read them all on one day, and they come back the next day and they reread them all, do they get the same answer? And the answer is that when we did this study, we actually got no deviations. The answers were exactly the same. Reproducibility is similar, except that the user changes from day to day. So now a new user has read the same set of samples, and then the same thing happens. When a new user read the same trays, they got exactly the same result. So I would definitely challenge anybody to run the culture method with the Petri plates to have that kind of performance. It just doesn't happen because colonies are much harder to resolve, and they're much harder to decide which are the proper ones or not. So that is a method against itself evaluation. We also did a lot of field trials. We had great success internally when we tested them in our laboratories, but of course, we're good at doing Legiolert, so we want to see if other people can do it too. So we did potable and non-potable water field trials with independent labs. We did in Germany and North America. I'm just going to go over North America today, but I'm happy to share the German results later if you'd like. All of the methods, we confirm them by secondary culturing. Now, I should mention that with Legiolert, you don't have to do any confirmations. You don't have to pull the sample out of the tray or do anything like that. Once you see the brown color, you're done. But in order to illustrate how specific the test was, we did do that for these trials. And this was a split sample, so the sample came into these labs. They split it in two and ran one with the standard culture method and one with Legiolert. And then we segregated the data for Legionella pneumophila. And the reason we did that is because the standard method does not have the capability of showing you just Legionella pneumophila. It sort of picks up a lot of pneumophila, and then it picks up a handful of other non-pneumophila too. So it can't really tell the difference. Legiolert is specific for Legionella pneumophila, so you don't have to do that. So looking at the statistics, this is kind of a busy slide, but it's actually pretty simple, and I'll walk you through it. The first trial we did was a potable water study, and we did this with a single laboratory. And there's my mouse here. There we go. What I want to focus your attention on is these graphs on the right. These are the same data on the top and the bottom. The top is zoomed out so you can see all the data points, and the bottom is zoomed in so you can see the lower concentration data points. And what you're looking at here is a plot where each sample got a concentration assigned by whether it was run with the standard method. In this case, we used the standard method's SM9260J, which is one of the favorites of one of the labs, against Legiolert. So every sample gets an X and a Y associated with one method or the other. Now, if the methods both give you exactly the same result, then all the points are going to fall on this middle 45-degree line. If the sample is more sensitive to Legiolert, it's going to fall above the line. If it's more sensitive with the other method, it's going to fall below the line. Now, in particular, if you zoom out, there was a few samples where Legiolert detected a very, very significantly high concentration of Legionella pneumophila, and the standard method detected almost none. These, I think, are the biggest problem with culture methods. This represents a situation where this is an actionable level here. You would do something about this in your water management plan if you got this result. The standard method is actually getting a result that's below the action level. This is the problem. If you zoom in and you look at all the smaller counts, I don't think these are really such a big deal. They're basically all on one or the other side of this 45-degree line, and they're more or less equivalent, but it's these big ones which I think are really the biggest problem. If you look at the statistics, we do a couple different statistics normally at IDEXX. Normally we do a standard t-test, which is good for most statistics, but in the case of these kinds of data, you have really big and really small values. That's called non-parametric data, which is t-test isn't really suitable for that. So we do another test called a signed rank test. And really what you're looking for is the stuff that's colored green. Whether or not something is less than 0.05 means that it is significant. If it's not less than 0.05, then it's not significant. So not significant by a t-test is significantly different in favor of greater sensitivity for Legiolert against this method. But I sort of couched it and said, well, because there's some different statistics going on here, let's just say that it's equal. I will tell you that in this particular study, there were no false positive signals reported. Every signal was actually confirmed to be Legionella in this study. So that's the potable water evaluation we did. We also did a lot of non-potable water evaluation. We did 4 different laboratories. And there's an interesting point I want to make here is that when we went out and we started talking to labs and said, well, we want to do some non-potable evaluations, can we run it against the CDC method? And they said, well, we don't really run the CDC method. Can we run our method? And we said, well, okay, let's do that. So we went to lab B and we said, well, we want to run this against the CDC method. So we don't run the CDC method. Can we run ours? I said, okay. Then we went to another lab and they said the same thing. And then we went to a 4th lab. And the 3rd and the 4th lab actually had completely custom homebrew methods that they developed. They were roughly based on the standard methods, but they weren't the same as any of them. So we just decided from a product evaluation standpoint, we would just test against whatever you got. And that's how we did the evaluation. So you're seeing the results against each of the individual laboratories here. And the layout of this is exactly the same. On the top, you have the zoom out, and on the bottom, you have the zoom in. And you can see that most of these things fall roughly on the 45-degree line or on either side of it. In a few cases, you have some pretty big points here that are above the line, which I think are those significant ones that I was talking about. This last method actually turned out to be a very, very good method. It was a little bit more sensitive than Legiolert, although Legiolert didn't really have too many failures to detect. It was really just a slightly lower sensitivity. So in general, Legiolert performed equivalent or better, with the exception of this last method. So if you look at the first laboratory, Legiolert was slightly more sensitive by the t-test, but not by anything else, and slightly less sensitive than this last method. What is really critical here is that you're looking at a real lack of green or red most of the places, which basically means that Legiolert is equivalently sensitive by and large. And that, in conjunction with the fact that it's so easy to use and so sensitive, makes it a really, really solid test for doing a risk management plan. This is really the key here. Another little piece of data here is if you look at the number of wells that we subcultured from, there were 671 wells. We pulled the material out and checked it to see if it was pneumophila. 652 times it was Legionella pneumophila, which is well below the accepted rate of 5% for false positivity. So this is the data that supports us calling this a confirmed test. So you don't have to do confirmation work. So let's say you do Legiolert on your water, and your test comes up as a positive result. Is this really bad? What's going to happen to me? The answer is not necessarily. There are other factors, and I've alluded to several of these already, besides presence absence. The big question is, how much is there? How much is some positive? The other key question is this a change from a previous test cycle? And the results from test to test have to be accurate if you're going to know this. If you just get anomalous results that show a spike, but they're not really a true reflection of whether there's a spike or not, that's not really accurate information, and it doesn't enable you to properly assess whether your water management plan is working. So this is just a very simplistic plot of a trending. You're seeing a limit here at the bottom, and your levels are fairly low, 5 mpn per 100 mil across the board, and all of a sudden you get this massive spike. This is what would trigger an action. And what you would do is you probably retest it, but If you trust that it's true, then you would add something here. Maybe chlorine, or you pump up your chlorine amount, or monochloramine, or whatever your favorite disinfection method is. And then after you add it, you would test again and make sure that whatever you've done has brought it back down to well below the limit. And that's basically how your risk management plan is going to work. So with that, I'd like to bring up my final thoughts. And this is just going to reiterate the things that I've already mentioned multiple times, which is that Legionnaire's disease is preventable with a risk management plan. The regulations and guidance and liability make it your responsibility to carry out that plan and make sure it's working. And there's lots of tools for you to do that. One key tool is an accurate test. Legiolert is an accurate, easy, and consistent test, which we think is essential for a risk management plan. It detects Legionella pneumophila, which is the primary clinical environmental disease agent. And it's an easy test, which means that more labs will offer it, and premise owners can react with the best information to protect the public as soon as Legionella pneumophila is found. So once again, I want to thank you. Again, I'm Dan Broder, and I work at IDEXX Water R&D, and I'm the team leader for Legiolert. And with that, I'm happy to take any questions.
Great, thank you, Dan. If you want to go ahead, if there's any questions, you can just type those in the question box and we can go ahead and take those. One question is, how common is Legionella in the Midwest?
How common is Legionella in the Midwest? Well, if you look back at the outbreak slides, you tend to see fewer of them in the Midwest. I don't actually know the full answer to that question. I would take a look at the epidemiological statistics in terms of what the CDC has available on their website and see whether or not there's a lot of incidences of legionellosis that happen there. I think in general it's a premise plumbing problem. So it can happen anywhere. I know the Midwest has pretty intense summers, so I would imagine that it's just as ripe for getting Legionnaire's disease cases there as anywhere else, but I can't say that I know that there's an increased or decreased incidence of Legionnaire's disease in that region. In general, outbreaks tend to happen in populous areas because cooling towers, if they're going to blow Legionella into the environment, if there's nobody around to inhale it, then nobody's going to get sick, or fewer people are going to get sick. So something like the New York outbreak probably was worse because New York is so dense. If you were somewhere that's a little bit less dense in the Midwest, then probably that's not going to be as big a problem. But I don't think Legionella itself is going to be present at a lower concentration in those areas.
Great, thank you. We have a few more questions. Are there any interferences with the test, such as copper or iron?
In terms of we haven't specifically measured the ability of copper or iron directly to affect the test. What we have measured is the performance against a lot of real water samples. And the reason why IDEXX tests work good is because we don't test them with artificial spike scenarios. We actually test them with real samples. So during the development, I actually contacted probably about 50 elite laboratories. So if you work in an elite laboratory, you probably have heard from me to try and get real samples that people are routinely processing in order to design the test to work against those. We haven't really experienced that as a problem directly. One of the reasons why I think it's probably not for the non-potable test, because you're only testing 0.1 milliliters of the sample in 100 ml final volume. So it's undergoing a 1 to 1,000 dilution. So if there is something in there that would have a problem affecting the test, it gets diluted out. A similar effect is found with potable water. It undergoes a 10-fold dilution, so not as aggressive. We're only using 10 mils of it. But as I said, we haven't seen any breakdowns in the performance of the test with real samples. So if there has been any iron or copper or anything like that in the water, we haven't seen it affect the test in a deleterious way.
Okay, great. And do you need to have any specific certification to run the test?
Yeah, so there are, to run the test, I have a slide that directly addresses this. Let me jump to that. Right, so for routine monitoring, A lab should be accredited by a nationally recognized accreditation program like AIHA or 17025 or TNI. They can also be elite certified. That's for routine monitoring. Legionella is not really designed for outbreak testing yet. The elite program brings on new methods into their scope every so often, and the next wave of that is going to happen in, we believe, in September 2017. Legiolert is already part of a pilot program that's occurring right now, with the intention of it then being suitable for elite certification. So this is what we would recommend for a testing facility.
Great, thanks. Kind of a follow-up to that. Do you recommend that only an accredited lab perform the test? And for example, do you need a precise incubation temperature?
We do recommend that a lab be proficient and have gone through a proficiency program in order to properly run samples. As far as the incubator is concerned, you do need an incubator that will hold the proper temperature. That's one part about it. Another part about the test is the test device itself, the Kewanee Tray, is a breathable device because Legionella pneumophila requires oxygen to grow. So that being said, the device itself will dry out over time if some sort of humidification is not provided. It's very simple to do. You just put in a water pan into your incubator, and that will provide enough humidity in order to keep the tray from drying out. So having the proper temperature and having the humidification in place is all you need to do. So if you're going to run both tests, you would need an incubator that is capable of holding both temperatures. So a 39-degree temperature and a 37-degree temperature are required to run both. If you're just running potable, then you can just have the 39-degree by itself, or vice versa for non-potable.
Great, thank you. We have a few more questions. In the standard method comparison, how does MPN compare to CFU, and how does the Legiolert limit of detection relate to the action level?
The MPN and CFU are directly correlatable. Both are estimates, actually. And actually, I have a slide for this as well. We work a lot with a statistician named Seppo Nimella. And, you know, we get this question a lot, and I'm not going to read through this, but the basic idea is that most probable number and cell-forming units are both estimates. They're just estimates because you're going to get some sort of Poisson distribution of cells in a given liquid sample. And whatever you deposit on your plate or put into your Kewanee tray are really just a reflection of whatever that specific local sampling you've done. So if you have 100 microliters out of 5 mils and you pull it out, it's just going to be what's in that 100 microliters. It doesn't necessarily reflect the true viable count. That being said, cell-forming units also is not a single cell for every colony. It's a cell-forming unit. So you could have 5 cells that are sticking together that if they drop on a plate, it's only going to look like one colony. So there's an area associated with that as well. The fact is that most probable number is a well-accepted mathematical algorithm that's been around for a long, long time. And in all of our studies, whenever we do a comparison from a plate count to the Kewanee tray, for example, with a spiking study where you take controlled introduction of a strain, put it into the Kewanee tray and drop it on a spread plate, and you measure the quantification of one against the other, they come out basically the same. So there's no functional difference. In addition, the EPA has approved our other test, Legiolert, which uses the MPN algorithm as well. So it's acceptable by the EPA. And Legiolert itself has been approved for a lot of different water matrices all over the world, which utilize the MPN calculation. Sorry, what was the second question?
I think you got all of it.
Okay.
We do still have a few more, though.
Okay.
With non-potable water, are sample treatment steps still required? Filtration and acid treatment?
The only step that's required for Legiolert is the fixed step of doing the pre-treatment. So let me jump back to that protocol slide real quick. All right, so for non-potable, every single non-potable sample is going to undergo exactly the same process. There's no deviation and no options. You would take an equivalent amount of your water sample and treat it with the same volume of the pre-treatment reagent which we provide, and that would be treated for a minute. and then that would go directly into the sample. Now, it looks like there's 0.2 mils that goes into the sample, but actually you're mixing it 1 to 2, so it's effectively 0.1 mil of the original sample.
Great, thank you. Another question, if I have a good chlorine and bromicide biocide control and also a non-oxidant biocide control, could I be sure that I also have good Legionella control even if I don't measure Legionella with tests?
No. You can, it's probably going to work, but the only way to confirm that it's working is to confirm that it's working.
Great. Okay, I think we might have one more question here. Perfect. I think that's all the questions that we have right now. Just to let everyone know, this session was recorded, and we will post this to the website a little bit later today and also send out a copy of the slides as well. So thank you so much, Dan.
Thank you, and thank you everybody for listening in. And if you wish to contact me, my contact information is directly in the slide deck. I'm happy to answer any more questions in detail if you have them. And once again, thanks for listening in.
Thanks, everybody.
Okay, bye-bye.